Very much. Do you hear me back there? Okay. So this is work absolutely in progress from Norway and from Oxford with Nick and Peter Hunter, and we're trying to expand this together with Dan Bird and Andrew McCulloch and Jim and other people in various directions in order to be able to achieve something. This is also a recruitment slide. Because, as you know, Norway is swimming in money, and they treat every professor, decent professor, very well, so this is my summer home. So, given this ambitious title and the fact that this is absolute work in progress, Golda Meir usually gives me some comfort, because she once told a visiting diplomat that, don't be humble, you're not that great. So this is the front page of the VPH. And considering what has been, I'm not promised this, some of you have promised this, that this progressive advance in computing power and associated information technology offers the potential of, that of course is the security belt, to deliver tailored clinical treatments based on simulation studies of the genetic profile of the individual requiring treatment. This is a thing very different from doing personalized modeling. Now we talk about going from the genetic profile of the individual, which is a tremendous challenge. So if we have promised this, we should better get going. And as you know, genetics is an old field, and it goes back to around 1905. Actually, where the term was first used in a letter from Bateson to Sedgwick. And within the same time frame, we got the genotype and the phenotype concept, and we got the gene concept. And to make a very long intellectual history short, these three persons are the main inventors of what we call quantitative genetics theory, and also population genetics theory in a sense. And this theory has developed into a very advanced mathematical structure, but it really boils down to, when we look at where its success comes from, is actually based on this equation. Here you see the difference between the offspring mean from one generation to the next, and this is the regression coefficient, and this is the difference between the parental average in the first generation and in the next generation, we select from. So this is the selection response, this is the heritability, and this is the selection differential. And what does it say? It says that it's based on the offspring on parent covariance. And this is the basic premise for natural selection to work. Had it not been such a relationship, there would be no adaptation in nature. So this whole mathematical structure is actually based on this relationship. And it doesn't need to be specific or be correct about all the genes the theory talks about in order to be correct about some of its predictions, because it's actually based on this relationship. So you can wonder, why do offspring resemble their parents? That's something for the lunch talk. But here at least you have a very simple equation. It's the simplest equation this week, I claim. So problems equal to explanatory ideals minus current capabilities. This is a good book, recommended. So here are some problems. Here are some terms: dominance, epistasis, penetrance, genotype times phenotype interaction, expressivity, heterosis, phenotypic plasticity, pleiotropy, canalization. And these are Google hits. I agree that some of these hits on this term may come from other sources, but still there are quite a lot of them relating to genetics. And these are problems, and they cannot be explained within the framework of neither population genetics or quantitative genetics theory. So Lewantin, many years ago, showed this very elegantly, that population genetics has lived in this field of the genotype space for 100 years, and quantitative genetics theory has lived in the phenotype space. So they deal very seriously with these guys. Deals very seriously with the genotypes and make a character out of the phenotype, and the other way around here. So what is really missing is, of course, the transition from the genotype to the phenotype. And of course, this happens all the time, I mean, in the evolution. So this is the Darwinian theater, of course, this is the genotype space. And if you're not able to make that bridge, we will not have any mature evolutionary theory, at least. So this is one of my hopes, that I'll be able to, together with my collaborators, to contribute to develop a quantitative genetic theory capable of linking genes, phenotypes, and population-level genetic phenomena through a causal understanding of the genotype-phenotype map. And actually, the first time I think the genotype-phenotype map concept came up was in Wonnington's symposium on Towards Theoretical Biology in '71 by Jim Burns. I will not, you can read briefly through this, but the point is that he uses the term, and he also defines really the research program we are dealing with today, but he didn't follow it up. So this is what I call a causally cohesive mathematical, or causally cohesive genotype-phenotype model. So there are mathematical models describing how phenotypes arise from lower-level processes in a causally cohesive way, and genetic variation is tied to those model parameters. And you get the population dynamic systems connected to genetic maps. This does not mean that you have to have every detail in place going from a gene to mRNA to protein and so on. This is a mistake. We don't need that to build very, very nice genotype-phenotype maps based on this type of model. But we need at least a term for describing models that are capable of taking us from genetics into the phenotype space relative to all other mathematical models. So this is the framework we use. We have spent several years on this. And so here you see you start from the offspring, you introduce your genotype-phenotype map through a mathematical model. You have genetics here, genes and markers and so on, and you have the offspring phenotypes comes out of this. It goes into the population. You have a selection there. You have parents. You have meiosis, and back again. In this way, you are able to combine the dynamic models with genetics, and with real genetics in the sense of genetic structures or genome. So here you can easily put in a real genome structure. So we have been doing this for generatory models for many years, and it works. We can certainly address a lot of phenomena by this. But of course, in order to be really able to deal with complex phenotypes, we have to enter the realm of multiscale modeling. And this is the reason why we are so interested in going in this direction. In order to get physics into it, in order to get traits where many genes contribute to the trait, and so on. Otherwise, we will not be able to address several of these phenomena. And this is just to make one point, is that when you go from genotype space, you have, of course, to parameterize that to phenotype space. But remember that every parameter here is a phenotype, which means that every parameter in these models have a GP map. And if that GP map is complex, you cannot just go from a sequence and say that you have a genotype data and you go to the phenotype. You have actually to understand the GP map or the parameter in order to parameterize that individual. So if we end up with needing to phenotype all the characteristics in order to make a personalized model, then we cannot go straightly from the genome to this. So hopefully, the GP map or parameters are mostly linear, they are easy to deal with and so on. Otherwise, we are up for some real challenges. Just to show you, this is a paper on salmon. And you should know in order to be a real man in Norway, you have to publish at least one paper on salmon. So this is my paper. So this is on the carotenoid dynamics. In this case, we built the dynamic model of the carotenoid dynamics and then introduced a GP map of the parameters. I was then able to actually to explain phenomena that had not been explained for 20 years in the literature by increasing the resolution of the model. And that is a quite simple model, so you don't need necessarily a dramatically complex model to deal reasonably well with GP map issues, combining dynamic models with genomes. Okay, so this is the work that has recently been done by Jon Olav and others. And so we took a model from Nick and Lirien, and based on the Bondarenko model, we have 8 biallelic loci, 6 determine 1 maximum current conductance each, and we had the NCX and the circa. And so we say we have 3 genotypes for each, coding for 50, 100, and 150% of baseline parameter estimate. And we run this for, so you get 6,561 parameter combination of trajectories. Of course, you get variation. This is from the calcium transients in cytosol, and as soon as you have variation, you have genetics. And this is a phenotypic correlation plot of this, and in some cases, and what does it represent is that in some cases you see, for example, here that it's quite well spread over, which means that there are all genes, almost all genes contribute to that phenotype. So in this case, you have the calcium duration in the cytosol up to 90% recovery, and the APD 50% reprioritization. And you see those two traits, they are not very well correlated, and they are spread well over. So there are many genes contributing to it. But if you look up here, you see you get a much more clustered thing, which means that the things start to get really structured. And here you see a phenomenon that is really important in biomedical genetics. You see that those two lower, you have much larger clusters here, which here you see that the homozygote for one gene here apparently confines the other genotypes to not have any phenotype, which is penetrance. So penetrance is an extremely complex thing for those in the clinic. And by this type of approach, I really hope that we're able to address the penetrance phenomenon in a decent way. And you can do a lot of various types of statistics on this, and you see, of course, these traits, they are certainly correlated, so they have a strong covariance structure, and you can do the principal component analysis of it, and you can take down those, if you take those just 3 of those phenotypes and take it down to from 21 statistics down to 3 principal components. And so that gives you a view of what is going on. And you get biplots, and here you have a PLS. So now you're not just trying to look at the variance among the phenotypes, but you have to also to relate actually to the underlying genes. So in this sense, we see that we can try to take from each other the different effects of the genes, the gene effects of the phenotypes from the genotypes to the phenotypes. So just to have one small here, and at the end of this to show where we're heading is that here you can plot the proportional variance explained of each phenotype by a given number of PLS components. So in this way you can see how much of the variance you can explain by those principal components from the PLS analysis. And you see, for example, for the circa, almost all the circa phenotypes can be explained by the first 2 principal components. And there are many ways to deal with this type of data. I'm not going to go into this. There's one point I would like to make before I end, is that age is still the best predictor of any common disease. And that is really bad for us, isn't it? That aging should be so difficult. Because unless we are able to handle aging in a decent way within the multiscale or in the physiome context, we cannot pretend to really deal with complex diseases. So what we try to do now is to set up a project where we try to address the aging heart from a modeling point of view. And I think we will learn a lot from that also about the normal functioning of the heart. Now we don't talk about the diseased aging heart, we talk about the healthy aging heart, because it will tell us a lot about what does it take to have a healthy aging in a sense of, and what does that mean when you increase your frailty over age in order to keep still the function. So I think that will also illuminate actually the normal physiological work to deal with aging. So Peter is on this, Nick and other people, and also Tom Kirkwood from Newcastle, very well-known gerontologist. So to end, This was written in 1997. The solutions to these problems, variation, heredity, selection, and other phenomena relating to evolution are in the first place statistical, in the second place statistical, and only in the third place biological. And we have, actually some people still think that, but we have moved a certain distance from this now, I think. And the GP map is a really central concept for all of us working with this. As soon as we talk about relating genes to phenotypes, we can deal with this GP map without having all details in place. And I think the cardiac physiome is one of the best places ever to really endeavor in this direction. Thank you.
Okay, so maybe 2 short questions, short answers, so we can get to coffee.
Can your formalism in principle deal with things like a knockout? And coming back to Dennis Noble, he gave the example of the NCX knockout, and one of the most surprising ones I've heard about recently comes actually out of Norway, the Circa knockout.
Yeah.
Can your formalism deal with something like that? I would think that's kind of the acid test for such an approach.
That's true, and but of course that relates also to that is more some sort of validity test of the current models of the single cell models. Are we able to reconstruct those Norwegian data? I mean, from Geir and Ole, and the Nick has been put on the case, and hopefully that. What it shows is that you have a temporal development, so you have no real phenotype for 4 weeks, and after 3 more weeks, the animal is dying. And of course, that is more a challenge actually for those of you who deal with the practical modeling of this. It's not a challenge to this conceptual or to this mathematical framework to deal with genetics. So we, in order to
the qualitative thing that should be.
Yeah, that's true, but to approach this, I mean, to deal with genetics is never truer than the actual state of the models you apply. But you're absolutely right. You should be able to handle that.
Very interesting. Is the genotype to model parameter like mini phenotype link based on experimental data? I didn't quite understand how you're doing that.
Maybe we can talk about that at coffee.